You might want to know the basics, which organelles are membrane bound and which ones are not. The structure of the plasma membrane. What all is present in the plasma membrane? Let me pull that picture. Here. What all is present in the plasma membrane?
Phospholipids, cholesterol,
Mm-hmm.
proteins.
Mm-hmm. Okay. So 70% of the plasma membrane is protein. That was good. What is the role of cholesterol in the plasma membrane?
Doesn't it help maintain the structure that's fluid? Like it keeps it fluid, I guess.
It gives a strength to the plasma membrane. So that membrane which has more cholesterol will be stronger. The one with less cholesterol will be more flimsy. And then the proteins, they all have different functions, lots of functions of the proteins which are present in the plasma membrane. Now, what is glycocalyx? Glycocalyx is the outer arrangement of the carbohydrate chains on the outer surface of the plasma membrane. Some of these carbohydrate chains are attached to the protein, so they are called the glycoprotein chains. And some of them are attached to the phospholipid heads, they're called the glycolipids. So all these chains which you see on the outside, that forms the glycocalyx, and that gives identity to the cell. Okay, so we've talked about, so you might want to know what are the different functions of these proteins which are in the plasma membrane. Now let's talk about the transport. What is passive transport and what is active? So first, what is passive transport?
Any transport that doesn't require energy.
Yes, no energy is required. And what is the direction of flow of the particles?
From high to low concentration.
Yes, from high to low. You have to remember these two, very important. Now, examples of passive transport.
Diffusion and osmosis, and simple and facilitated diffusion.
Yes. So we have simple diffusion, we have facilitated diffusion, and osmosis. Okay. And active process is exactly the opposite, and we'll talk about that in a minute. So, what is happening in simple diffusion? Give me an example of simple diffusion.
Could be like putting dye in water and watching
Yes.
the water or the dye spread out.
Okay. And in the body, do you remember any example happening in the body? Simple diffusion. Here. The movement of oxygen, exchange of oxygen and carbon dioxide in the lungs is by this process of simple diffusion. What is facilitated diffusion?
Small charged and polar solutes, they're assisted by membrane proteins, channel mediated or carrier mediated diffusion to go into through the cell membrane into the cell and out. And out.
Okay, so that means you need to have a membrane protein or a carrier protein for the movement of those particles. Any example? We did that on the other day in the lab. What did we use for
The sodium, potassium, no? Perfect.
for facilitated diffusion? What did we use the other day? Glucose. Glucose amino acids are two very important molecules which require a transport protein. So that is facilitated diffusion. Does it require ATP?
No.
No, because it's a passive process. Now, what is osmosis?
Movement of water.
It is movement of water across a selectively permeable membrane. And when we say selectively permeable, that means that membrane will not allow water, solutes or the solids to pass through, but it will allow only water to pass through it. That is osmosis. Now, what will happen? You know the three types of solutions: hypotonic, hypertonic, and isotonic solution. So, what is isotonic solution?
Equal number of solvents in the cell and the solution.
Okay. Let's say equal concentration of solutes in the cytosol and in the solution. The concentration is the same. The solutes may be different because you can create any solute, like we said, sodium chloride. 0.9% sodium chloride is isotonic solution. That means this 0.9% sodium chloride has the concentration of this is same as the concentration of whatever stuff we have inside the cell. It doesn't mean that inside the cell also has same sodium chloride. No. Inside the cell we have potassium, we have negatively charged proteins, we have little bit of sodium also. So all those things put together, that concentration is same as 0.9% sodium chloride solution. So that is what is isotonic. What is hypertonic solution? What is hypertonic?
When the cell shrinks because water is leaving the cell.
Sorry, say that again, please.
The cell shrinks because water is leaving the cell.
Okay, but what when you say it's a hypertonic solution, is this solution more concentrated or less concentrated than the cytosol?
More concentrated.
Yes, it's more concentrated, which means it has less water. So that's why water will start leaving the cell and go out, and the cell will shrink and die. Crenation. Hypotonic solution is the opposite. The solution is less concentrated than the cytosol. That means it has more water. So since we have more water on the outside, the water will start going into the cell and the cell will again burst and die. This is the stuff you might want to really know. And for some reason, physiology people love to ask you questions about this tonicity. And I've taken lots of exams, and whenever I took an exam related to physiology, no matter what, there was always a question related to this tonicity, which is easy to answer once you understand what's happening. It's not difficult. Because this directly is related with the clinical scenarios. When you have dehydrated patients come, you have to figure out what kind of solution you want to give, or when somebody is overhydrated, what kind of solution you want to give, or if you want to give any solution. So it depends a lot on the patient's condition, what solution you want to give. Okay, now active transport. What are the characteristics of active transport? What is the direction of flow?
Against the concentration gradient.
Yes, against the concentration gradient. That means from low to high. Is ATP utilized or no?
Yeah.
Yes. Give me one example of active transport.
Sodium and potassium.
Sodium potassium pump, which is located in the plasma membrane, is an example of the active transport, and which is the primary active transport. And we did that on the other day in the lab. 3 sodiums is to 2 potassiums. The sodium potassium pump is going to pick 3 sodiums from inside the cell and throw them out. That's where sodium belongs to. And it's going to pick 2 potassiums from the outside and throw them back into the cell, because that's where potassium belongs to. And it will utilize energy, ATP, when it's doing this. I hope the lab which we did last week will help you in all this, because you physically did it in class. Okay, and now the other type of examples of active processes is the vesicular transport. And vesicular transport includes exocytosis and endocytosis. What is exocytosis?
It
Move
moves
substances out of the cell.
Yes, so things are moving out of the cell, and that is exocytosis. And what is endocytosis? What are the 3 types of endocytosis? The first one is here, phagocytosis, which is you have a particle outside, a solid particle, and the cell is going to eat that particle. That's why it is called phago. Phago means eating. And then pinocytosis is when there's a fluid outside and the cell will take that fluid in. That is pinocytosis. It's same like phagocytosis, except that it's not a particle, it's a liquid. And then we have the receptor-mediated endocytosis, where the particle is going to first attach to the receptor and then the cell will take it in. So that's why it is called receptor-mediated endocytosis. And here is a summary. You might want to look at this one for a review and add the examples to it. The next thing is the membrane potential. What is the resting membrane potential? You have the definition here. It's the electrochemical gradient of the cell when the cell is at rest. That means it is not transmitting any signal. especially in terms of muscle cell and the neuron. The cell is at rest, then the electrochemical gradient across the plasma membrane, that is called the resting membrane potential. What maintains this resting membrane potential? What maintains it? Sodium potassium pump. That is the job of sodium potassium pump, is to maintain the resting membrane potential. Know the three definitions. What is depolarization? Here. We are at resting membrane potential minus 70. This is for a neuron. Depolarization is when the inside of the neuron starts becoming positive. It starts going towards the positive side. You see that this is the positive side. That is depolarization. Repolarization is when it's after depolarization, it starts going back to the resting membrane potential. That is repolarization. And hyperpolarization is when it from the resting membrane potential, it becomes even more negative. That is hyperpolarization. Please know these three definitions. You will need this information a lot in the next few chapters. I will keep talking about depolarization, repolarization, hyperpolarization. So know these definitions now. Okay, and then this is the stuff you can read on your own. The organelles and their functions. And this is your work, the lab worksheet from last time has all this. So you can read through this. Membrane bound and non-membrane bound, everything. Then what are the types of cell junctions? See, I've skipped those slides. That doesn't mean it's not there on the test, but that's something you guys have done and you can, that's just for you to memorize. What are the three types of cell junctions? Tight junctions, the desmosomes and the gap junction. Please know these three. Tight junction is like staples between the two adjacent cells. So here you have one cell, you have the other cell, the two plasma membranes, they are stapled at intervals. That is a tight junction. Desmosome is you have a protein plaque. It's like here, both sides there is a plaque, and in there are kind of chains going across from one plaque to the other. So keeping the two cells close together. That is desmosome. And then gap junctions is there are tunnels between the two cells. So from one plasma membrane to the other, there's a tunnel and things can go across that tunnel, and that is called the gap junction. Okay, and that's the stuff here. Okay. Any questions?